You are listening audio paper series with Yasir, and today's guest is Professor Mark Weber from Quadram Institute, Norwich, UK, to discuss one of his publications, long-read sequencing for identification of insertion sites in large transposon mutant libraries. It is published in Scientific Reports. Welcome, Mark.
Hi, Yasir. Good to see you again.
Nice to see you. Why was there a need to use long-read sequencing to identify insertion sites, as there's already a method to identify transposon insertion sites?
So, as you say, the current methodology is well established to identify the positions of transposon inserts in the mutant library, and that largely relies on using the Illumina platform, which provides short reads. And that works very well, provides a lot of information. However, there are some bits of the genome which are hard for short reads to map accurately. So what we wanted to do was try and use some long read technology to get us the ability to identify those areas of the genome where there are inserts.
Right. So you mean there are certain areas in the genome where the short read sequencing doesn't work, or we cannot identify the insertion sites with confidence?
Exactly that. So about 10% of most bacterial genomes are what we call a repetitive element. So it's a piece of DNA that's somewhere else in the genome as well. So that might be a ribosomal operon, which is there's lots of copies. Mm-hmm. Or it might be an insertion sequence. Now, a short read that maps inside one of those, you can't read out into the flanking genome, so you don't know which position in the genome that insert's at. So you know it's in one of these elements, but you can't accurately tell which one. And sometimes that might be biologically important, and we're missing that information at the moment with the short read method. That's why we wanted to try, and that's one of the reasons, we wanted to apply long read sequencing.
Okay, that's great. So how did you plan to do this long read sequencing from transposon insertion sites?
Okay, so we're very lucky at the Quadram that we have a very strong sequencing team and informatics team, and we use various sequencing platforms a lot. And we use the Oxford Nanopore long read platform for lots of applications in the institute. So we adapted our protocol to essentially generate longer fragments of DNA in the library preparation section, and then some adjustments that we mentioned in the paper about the sequencing methodology to make it compatible to run on an Oxford Nanopore sequencer, but nothing too dramatic to do that. So we essentially used our normal process and adapted it to run on the Oxford Nanopore instrument.
Okay, so you used Oxford Nanopore ion cells to sequence it. Cool. So what was the major finding of this study or this method?
So a few things really. So the first thing was that with does it work? Can you do it? And yes, you can. So it works. You can map long reads. I mean that's conceptually no surprise really that you should be able to, but we showed that you can. You can also generate enough information, enough long reads. And then when we compared the outputs of the data from the short read platform and the long read platform, they were largely comparable. So if you look at the position of inserts, the number of inserts, the number of essential genes that each method produced, they overlap very well. So that tells us with confidence that actually the long read method works, and it gives equivalent data for most of the genome to short read data.
Cool. So can you give an example of repetitive elements that you manage to resolve by using this long read sequencing method?
Sure. So obviously the first thing we did is just try and check that it works, that it was comparable to the previous kind of data for most genes. But of course, we're interested in those repetitive elements. So we looked at some of those. We looked at the ribosomal operons, and we looked at some insertion sequences that are we did this with E. coli in an E. coli transposon mutant library. And we found that we were able, using the nanopore data, to unequivocally map inserts to one position in the genome, rather than saying this insert is in one of the rRNA operons, we don't know which one. We can say, no, it's this one. And that gave us confidence that it's actually done what we wanted it to do, that it works.
Cool. That sounds great. Okay, so what are the applications of this method where we can use it?
There's a couple of things. So firstly, being able to use the Oxford nanopore ion sequencer is actually quite useful for various ways, because you don't need to commit an entire Illumina flow cell to running a data set. So actually, it gives you a bit more flexibility. It doesn't generate as much data pound for pound, but it gives you enough. So you have scalability in your experiments. If you're doing a small transposon mutant experiment, then actually Oxford Nanopore is a potentially viable solution for sequencing it. So that's one thing, with all the flexibility that that entails in terms of access to the instrument, is very easy. But secondly, and the real interest in this for us was to understand some biology about saying actually these these repeated regions of the genome are important. So as I said, kind of maybe 10% of a bacterial genome is currently blind to the traditional transposon mutagenesis methods because we just can't say where in the genome these inserts map, which is the whole point of using transposon mutagenesis. So now actually we're able to get some more biological insight to say, are all these transposon mutants being enriched in one insertion sequence that's in a particular part of the genome that affects the flanking gene content, for example? So now we can actually start to use this method to study biology and get to that kind of final 10% of the genome that we're missing.
Lovely. Thank you very much, Mark, for your time and sharing very interesting research paper.
It's my pleasure, Yasir.